Okay, hello. Today we're talking about, we're going over chapter 8 in crop production. This chapter deals with plant physiology and growth. And one process that's necessary to plants for growth is photosynthesis, and that's the process by which plants capture energy from the sun and convert it to chemical energy. The equation for photosynthesis is shown below. It's also in your written notes and in your book. Just basically carbon dioxide plus water with energy from the sun gives you that C6H12O6 is glucose, which is the most common sugar used for energy, and plus oxygen. And photosynthesis happens in the leaves of plants. This is a cross-section of a leaf. Plants take in CO2 through the stomata. They're just little holes on the bottom, which are have guard cells on the outside that open and close them. And photosynthesis happens mainly in the leaf cells called mesophyll. They're the cells kind of right under the top layer that are kind of like oriented in kind of an up and down way. And the mesophyll just means middle leaf. And inside of the mesophyll cells are little organelles called chloroplasts. And this is just a kind of a more of a blown up drawing of a plant cell. And the chloroplasts, which are the little green structures scattered throughout, they have a semi-fluid substance called stroma and little stacks of disc-shaped sacs called thylakoids. You can see them on this drawing. They're kind of look like little stacks of green coins almost. And the thylakoids have light absorbing pigments in them. And the main one is chlorophyll. And that absorbs violet, blue, red light, but it reflects green. And there's also, chlorophyll is the main pigment of photosynthesis. And there's also accessory pigments that absorb other wavelengths and transfer the energy to chlorophyll. And they, those are different colors. Some are yellow, some are purple, some are red. But normally you can't see the color of the accessory pigments because it's masked by the chlorophyll through most of the plant's life until fall when the chlorophyll breaks down because the plants are no longer actively doing photosynthesis, so they don't need chlorophyll. And that's why then at that time you can see the colors of their accessory pigments. So some leaves, depending on the species, will turn yellow, some red, some purple, orange in the fall. So that's the reason plants' leaves change color in the fall. And then, like plants, the photosynthesis is made up of a light-dependent phase and a light-independent phase. They used to call it the light phase and the dark phase, but actually the light-independent phase doesn't have to happen in the dark. It just doesn't need light. So the light-dependent reactions take place in the thylakoids, and then solar energy is transferred to energy carrier molecules. And in this phase, water is consumed and oxygen is released into the air. And then the light-independent reactions happen in the stroma, the fluid part of the chloroplast. And energy is transferred from energy carrier molecules ATP and DPAC to glucose, which is sugar. And the glucose is manufactured from CO2 and H2O with enzymes that are present in the stroma. And the process of glucose production is a series of reactions known as the Calvin-Benson cycle. We're not going to go over the whole cycle here because it's kind of complicated. And most of the glucose is immediately converted to sucrose, which is kind of like a disaccharide. It's like a form of sugar. Sucrose is actually table sugar. It's the form that we're most familiar with that we eat and put in our food. Or else starch. Starch is just a long chain of sugars that's like more of a long-term sugar storage for plants. And the starch grains are stored in the stroma for when the plant needs them later. And later on they'll be converted to sucrose for transport to the leaves, stems, other parts of the plant. And then the depleted energy carriers just return to the light-dependent reactions and they're re-energized and used again. And the plants have different ways of, harvesting CO2 and fixing, The process of harvesting CO2 and making glucose is known as carbon fixation, and there's different ways that this happens based on the plant and the type of climate it's evolved under. So the C3 pathway of carbon fixation and making glucose is used by typical plants of temperate areas, areas where it's not too hot or too cold. And those, as far as crop examples, those are beans, rice, wheat, and potatoes, and some others. So these are basically plants that evolved in areas where the temperatures aren't too hot or cold. And it's all trees also are C3s, most trees. And it's named because the most important molecules in it have 3 carbons. And in this case, carbon fixation and glucose manufacture both take place in the mesophyll cells of the leaf. And the problem with this, it works well in temperate areas, but it doesn't work well in hot, dry weather. And that's why these types of plants don't do as well when it's hot and dry. So the water supply is adequate, plants have to open their stomata to let in CO2. But when water is limited, plants close their stomata to try and conserve water. But when they're closed, they can't take in CO2 or release O2. And in that case, the molecule that normally combines with CO2 in photosynthesis combines with O2, and that's a useless process called photorespiration. It doesn't do anything for the plant. So some plants in areas that have evolved in areas that have strong sunlight and hot temperatures use a process called the C4 pathway. And these include many grasses like corn, sugarcane, sorghum. They've evolved this different pathway that allows them to do better in hot, dry areas. And in this case, carbon fixation happens in the mesophyll, but glucose production happens in the bundle sheath cells that are around the veins of the leaf. And then after that happens, the normal C3 cycle will happen. And it's called the C4 plant because the initial molecule in carbon fixation is a 4-carbon molecule. And because of this different process of carbon fixation, these plants can do better in areas that are warmer and tend to go through dry, hot area cycles. Then there's some plants that have evolved in really dry, hot areas like deserts, and they store water in fleshy leaves, or some, like the cactus, their leaves have actually become needles, and they do photosynthesis in their stems, in the fleshy parts of the plant. And in these cases, they fix carbon and produce glucose in the same cells, but in 2 stages, one at night and one during the day. And so during the night, when it's cooler, they keep the stomata open and make 4-carbon molecules that they store in the vacuoles, which is just the kind of big water sac in the center of each cell. And then during the day, when it's hot, they close their stomata, and the organic acids that they made during the night are broken down and release CO2. And that's the C3 cycle happens there. So in this case, the plants have just evolved so they're able to keep their stomata closed during the day when it's so hot they would lose too much water if they had them open. And then once the plant has made the sugar and needs to use it, the sugar needs to be converted the sugar formed in photosynthesis needs to be converted to energy for metabolism of living cells. And so that process is called respiration. And you can see, if you remember the equation we put up before, the equation for respiration is exact opposite of the one for photosynthesis. The glucose and oxygen are broken down into CO2, water, and chemical energy. And so this happens in plants when they need to, use the sugar they've produced for energy or growth. And it also happens in us when we eat a plant or eat sugar. And so the first stage of respiration is called glycolysis. That just means breaking sugar. It takes place in the cytoplasm. And so the glucose molecule is broken down into 2 3-carbon molecules, and this releases some energy that generates two ATP molecules. Glycolysis doesn't require oxygen. It works the same way whether oxygen is there or not. But what happens next depends on whether or not oxygen is available. And if the oxygen is available, the pyruvate enters a second phase called cellular respiration, and that takes place in the mitochondria, the little powerhouses of the cell. And this is a lot more complicated than we're showing here, but for the purposes of this class, you don't need to memorize all the little steps here. But so each pyruvate is split into CO2, which is released, and then a molecule called acetyl-CoA that enters a series of reactions called the Krebs cycle or citric acid cycle. And the Krebs cycle is actually about 13 steps, which, like I said, we don't need to memorize them all. But it's called a cycle because it's a series of reaction, and the product of the last reaction just feeds into the first, and so it starts all over again. And the Krebs cycle produces 2 ATP molecules and then donates energized electrons to energy electron carrier molecules. And then those donate their electrons to what's called the electron transport chain. It's just a little series of enzymes inside the inner membrane of the mitochondria. That's why the mitochondria on the cells are called powerhouses, because this is where most of the energy is generated. And then the electrons moving along the chain, develops an energy gradient and pumps hydrogen ions into the inner membrane of the mitochondria. And this is where most of the energy is developed, 32 to 34 ATP molecules, depending on the type of cell. And so at the end of the electron transport chain, oxygen is needed to combine with 2 hydrogen ions and form water. And if oxygen isn't available, the electrons pile up in the chain and the process comes to a halt. That's why most living things can't survive without oxygen, including us. And in those cases, even, plants, you normally hear of plants taking in CO2, which is their main, that they need for photosynthesis, but for respiration, they also need oxygen, just like we do. And that's why if a plant is, like plant roots especially, or like the areas, if they're growing in an area that's deprived of oxygen, they don't do well either. And then there's also a process called transpiration. It's just the water loss from the stomata of the plants. Like I said, when they have them open, they lose water. And this, is a good process to some extent unless they lose too much water, because it cools the plant, moves nutrients throughout the plant, and also aids in uptake of minerals. And so water moves through a plant along a concentration gradient. A concentration gradient is just a area where there's a difference between high and low concentration. So the water moves from the roots, which is an area of high concentration, to the leaves, which are a lower concentration. And then, the excess goes out the stomata. But if there's not enough water to support transpiration, the plant will close its stomata, trying to conserve water. And eventually it'll wilt and die if it's really lacking water. And this whole process is affected by soil moisture, air temperature, things like that. If it's hot and windy, the plant tends to lose water faster. And then evapotranspiration is just the plant's total water use. And that includes all the water that is lost in transpiration, but also the water that's evaporated from the surface of the leaves and from the soil around the plant. And the ET, or evapotranspiration ratio, is just the weight of water required to produce the crop's dry weight, the total dry matter weight and of the plant during the year. The total plant growth it depends on type of just basically it's the amount of water needed to produce the total crop plant for the year. And that depends on whether it's a C3 or C4 plant. C4 plants can get by with less water. And then also the number of stomata, hairs on the leaves, and waxy coating on the leaves, which influence the amount of water that's taken in or lost. And C4 plants are the most efficient at using water. That means they are able to produce the most plant matter for less water, while C3 legumes are the least efficient. And this little table is right from your book, but it just shows typical evapotranspiration values for different Midwestern crops over 1 year. And you can see that in order to produce alfalfa, a certain amount of alfalfa, you would need more water than something like alfalfa, which is a legume. you would need more water than things like dry beans and winter wheat, which use the least amount of water to produce an equal amount of plant material. And then some plants are capable of what's called symbiotic nitrogen fixation, and those are the legumes, which is the family Fabaceae. And that can that includes things like alfalfa and also beans, peas, soybeans, things like that. They are able to convert atmospheric nitrogen to a form plants can use. And this involves a symbiosis is just when two things live together, two species live together and both benefit. And it's a partnership between legumes and species of bacteria called rhizobia. There's a different rhizobia for each species of legume. And so just what happens is there's a bacterial enzyme called nitrogenase that the rhizobia use to convert nitrogen from the soil into ammonia, which is NH3, and that's changed into ammonium, which is used by the legume plant to form amino acids and proteins. So basically it's taking the nitrogen, the N2 nitrogen, and turning it into a form that plants can actually use. And so the rhizobia infect plant root hairs, find little extensions of the roots, and they stimulate development of nodules on the roots. And the process of nodule development is shown in this diagram, which is from your book. And then, it's able, the bacteria get some sugar from the plant, so they benefit, and then they give nitrogen to the plant, and the plant benefits. And then photoperiodism is just a plant's reaction to changing durations of light and darkness. Photo means light, and period is just, how long, basically how many hours of the day are light and dark. And it influences a lot of different reactions and developmental processes in the plant. And it involves the pigment phytochrome, which is it's inside the plant, and it senses changes in amounts of red light, which is just an absorption peak in the wavelength spectrum of 660 nanometers, and then far red light, which is an absorption peak at 730 nanometers. So phytochrome has two forms, PR and PFR, and the active form is PFR, which is located in the vegetative portions of plants and seeds. And the phytochrome is normally in the cytoplasm of a plant cell, but when it's activated, it travels into the nucleus, and then it induces changes in vegetative or floral cell production. So when phytochrome is activated, it will influence the plant to go into a different stage, either maybe start producing flowers and seeds, or to go dormant, or whatever, just depending on the situation in the plant. And plants have different flowering responses to day length, the amount of light or the amount of darkness in the day. And the little chart here, which is from your book, just shows the difference in different plants, and different stages, basically the influence of day length. And so there's some plants that are called short day plants, and they flower when the days have become shorter than a certain time or photoperiod. And those are late summer or fall flowering plants or short day plants. So when the days have gotten as long as they're going to get and they start getting shorter, then the short day plants will flower. Long day plants flower when the photoperiod is longer than a certain time, and those would be spring or summer flowering plants that as the days get longer and longer, when they get to about June and July, when the days are as long as they're going to get, then those plants are stimulated to flower. And day neutral plants don't flower in response to day length at all. They just flower when they reach a certain age, or maybe when they've gotten to a certain level of growth, but day length doesn't influence them at all. And then dormancy and germination, there's different responses in seeds, based on time of year and shortening of days and whatever. When plants in northern latitudes respond to shortening days and decreasing temperatures by going dormant. And dormancy you think of as a time when there isn't much growth going on, but actually there's a lot happening in the plant that's involved in getting ready for winter. And so like maybe the plant doesn't seem to be growing much, but it's actually storing energy in the roots, maybe moving sugar into the root areas and forming crown buds so it can regrow in the spring. And then so like seed germination responses, deeply buried seeds that are not exposed to light will not germinate, but those are nearer the surface that receive enough light will germinate. And light in a germinated seed, once again, is received by phytochrome, and then that will activate a vegetative growth in the plant towards the soil surface. And some plants that are evolved in colder areas need to go through a process called vernalization. And for example, like winter annual small grains, in order to flower in the spring, they require exposure to a short photoperiod and low temperatures in the winter. So basically, if they're not exposed to winter conditions, they won't flower. And that includes like winter wheat and winter rye that are planted in the spring. If they wouldn't go through the cold period and short days of winter, they wouldn't flower. Like if they were planted in an area that was just hot, that didn't really have a season change, they wouldn't flower in the spring. And then differences between growth and development, they are sometimes used interchangeably, but they actually have two different meanings. Growth is the irreversible increase in number and size of cells, and so it's just the plant is getting bigger, it has more cells. And development is a process of changing to a different maturity or growth stage, and like going from a seedling to a mature plant, or starting to flower, or seed maturation is just development is going to a different stage. And then there's different ways to measure plant growth. It can be measured as fresh weight or dry weight. Fresh weight is just like you would cut down or harvest the whole plant and, weigh it exactly as it is before it dries. And then dry weight is, drying it down and measuring just the dry plant material. And you can also measure height, length, leaf area, things like that. And dry weight is sometimes used to compare from one area to the other the actual plant growth, because you basically just dry down the plant and measure the plant tissue minus the water. And that way you know exactly how much plant tissue is there, because the weight of the water, if the plants in one area are, have enough water and the plants in another were dry, it might make it look like there actually was more plant material than there was. And agronomists and soil scientists use what are called growing degree days, which is just an equation that's developed. It's an index of heat accumulation over time that's developed to estimate how crop development will progress in response to temperatures. And basically this is used because, like, if you have a variety of corn or whatever that's supposed to mature in 60 days, 80 days or whatever, it's not always going to be 60 days or 80 days every year, because if you have a cool year, it will take longer for your plant to mature, or if it's warmer, it will be, it will mature faster to a point. And so the equation for growing degree days is just, this would be for each day. You take the maximum temperature and the minimum temperature for the day, just the high-low temperature for the day, add them together, divide that by 2, and then you would subtract a base temperature, and that's different for each species. A base temperature is just the temperature below which no growth will occur. And for plants like corn and soybean, the base temperature is 50 degrees Fahrenheit. So if the temperature for the day is below 50, if the temperature is below, maximum temperature is below 50 degrees Fahrenheit, the plant will just sit there. It won't really grow any at all on that day. But also in some plants like corn and soybean, temperatures above 86 don't result in more rapid growth. It's like if it's really hot, like 100 degrees or whatever, that plant isn't going to grow faster. It's just going to sit there and kind of be dormant for that day. So in that case, when you do the equation, you would take any temperature above 86 would be considered equal to 86, and then so the degree days are just figured for for each day, and then there's like scientists have determined approximately how many degree days a plant will need to get to different stages like flowering, seed production, and whatever. And that's just a way that you can use to kind of predict what stage your plant is going to be when it's planted. You start developing growing degree days from the day of planting on, and you can determine about where your plant is going to be by the development of degree days. And people evaluate crops and also weeds by growth stages, which they're named and by different scales. Like this is a common growth stage system for corn. By you would look out and say, well, my crop is in the VE stage if it's just at the emergence stage, and this shows what each stage kind of looks like. And there's different systems online you can look at, which shows the plants in more detail. So you could look at each plant and determine what stage it was in. Like the V1 stage has the first true leaf, and so on. And then there are the R stages for corn, or when it's actually developing seeds, kernels, and you can look at the kernels and determine which stage it's in. And there are similar growth stages in your book for wheat. There's a couple systems for wheat. There's also one for soybean based on, whether it's vegetative, how many leaves, flowering. And there's one for alfalfa too. And we'll look at, have some assignments later on that will, look at determining the growth stage of plants. And this is important because for herbicides, they often will say, apply before a certain growth stage or after a certain growth stage. And in order to do that safely, you would need to know what growth stage your crop was at. Like if it might say, for example, apply this herbicide after the 3-leaf stage, you would have to go out there and make sure you know what stage your crop is in, because if you would apply before that, you might potentially injure it. And sometimes they'll mention stages of weeds as well, that maybe it won't kill the weeds after the seedling stage or something like that. So growth stages are important to know. And then plant life cycles, just there's different crops and weeds that go in. Like the life cycle just tells how long they have to complete their life cycle in 1 year, based on how many years or growing seasons. And different species go along with different ones, and also different weeds too as well. And there's lots of other examples. I've just got some major ones here. So an annual completes its life cycle in 1 year, 1 growing season. And there's summer and winter annuals. Summer annuals are planted in the spring, grow during the summer, mature in the late summer to fall. And so crop examples of summer annuals would be like corn, soybean, spring wheat, and rice. And weed examples are pigweed, velvet leaf, foxtails, ragweed. There's lots of others too, but these are some major ones. And so it's important to kind of know what your life cycle of your crop is, as well as some of the weeds that you might be trying to control. And a winter annual is planted in the later summer to fall, and it just grows vegetatively before winter, and it overwinters in a dormant state and flowers in the spring. And crop examples would be like winter wheat and winter rye. And there's some winter canola varieties now. And then weeds that are winter annuals are chickweed and wild mustard. And then a biennial needs 2 years, 2 growing seasons. They grow vegetatively the first season and accumulate reserves for the winter. In the second year, they grow back, flower, set seed, and die. And crop examples of biennials are sugar beet, sweet clover, and carrots. And weeds include wild carrot and bull thistle. And then perennial persists, keeps growing for 3 or more growing seasons, depending on the climate. There's some that might be longer-lived perennials in, a better, warmer climate, and in a colder area like North Dakota, they might be a shorter-lived perennial. And they might flower each year, but they also grow vegetatively throughout their lives. And perennials will have some specialized energy storage organ, usually such a taproot, rhizomes, which is like an underground stem, or a corm, which is kind of like a bulb of like a crocus or something like that, or like corms. That part continues to survive. And perennial crops include things like alfalfa, clover, switchgrass. You harvest the alfalfa every year, but it's still a perennial. And weeds would be quackgrass, dandelion, Canada thistle, leafy spurge, and others. Perennial weeds are often hard to control just because you can't always get the vegetative structures under the ground. And some plants are treated as pseudo-annuals. That means, pseudo means false, and they're not really annuals, but they're treated as annuals when they're grown as crops. For example, like sugar beet, we said is a biennial, but we don't let it grow for 2 years when we harvest it for the sugar. We plant it and harvest it at the end of the first year. Harvest the beets at the end of the first year. If it was allowed to act as a biennial, which it naturally is, it would grow back the second year and flower and set seed. And cotton is also a perennial. It's treated in cropping systems as an annual. It's harvested and planted and harvested every year. And alfalfa and red clover are perennials that are grown as annuals for green manure. And like you would plant it and then and then plow it down later. So it's actually treated as an annual even though it's a perennial. Okay, that's all for this chapter. If you have any questions, please let me know.